The virus is the smallest infective organism, microscopic, and which can only replicate inside the host cells. In the environment, it's dead, but as soon as it enters the host cell, it hijacks the pathway of metabolic pathway of the host cell and try to multiply as early as possible. And there is initially a viremia, and each and every virus is practically tissue tropism is there, and on their specific tissue they produce the lesion. And the main thing is that, like bacteria, it is not visible. So we need the help of microscope to observe it. It is very difficult to cultivate. No synthetic media is available. It is very difficult to maintain. And treatment is also limited. So clinician often faces problem because of the overlapping signs and symptoms of the different type of virus, especially within the group of respiratory tract, within the group of gastrointestinal tract, or virus producing CNS lesion, often produce these overlapping signs and symptoms, and it is very difficult to diagnose. But now the recent advancements of the microbiology and virology, there are PCR, but the scope is limited. So the clinical findings, clinical features, and the initial identifying feature of patient is very important. So let us discuss one by one very quickly. If you see the history, the Russian scientist Ivanovsky, 1892, he first declared that this is something is there, but the name is not known at that time. But some microorganism or something is there. And later on, the infective nature was demonstrated by Adolf Meyer in the tobacco mosaic virus. Later in 1898, the Dutch microbiologist Martinus, he confirmed a new type of infectious agent, contagious living fluid. The name is not known. The first animal virus isolated, foot and mouth disease virus in animal in 1898. But the first virus in human was yellow fever virus in 1901. The bacteria first discovered in 1915 by Trott. So the first virus isolated is yellow fever virus. The virus differ from bacteria. This is almost known to you. Obligate intercellular, possesses either DNA or RNA, but never both. Smaller than bacteria, can be passed through the bacterial filters. Cannot be grown on artificial cell-free media, grown in animals, embryonated egg and tissue culture. Do not have a proper cellular organization, lack the enzymes necessary for protein and nucleic acid synthesis, as it hijacks the pathway of the host cells, and not susceptible to antibacterial or antibiotics. The morphology, the entire virus particle is called, the infectious particle is called virion. The infectious particle or the whole virus is called virion. It comprises of nucleic acid, DNA or RNA surrounded by a protein coat called capsid. The protein coat called capsid. Some viruses also have an outer envelope. The largest virus is smallpox virus. Smallpox virus is the largest virus. And the smallest virus is parvovirus. 20 nm. The nucleic acid may be DNA, maybe RNA, maybe double stranded, maybe single stranded, maybe segmented, maybe unsegmented, maybe circular, maybe linear. So these are the variations of nucleic acid of the virus. I repeat, segmented, unsegmented, circular, linear, single stranded, double stranded. With the capsid, it is viral origin. The capsid is viral origin. Outer to this particular capsid, in cases of some virus, not all, there is a layer that is enveloped. That is lipoprotein in nature. Capsid, viral origin, protein in nature. And the outer wall is lipoprotein, lipid and protein. Why? Because during budding from the host cell, it acquired the outer wall of the host cells, that is lipid in nature. And the protein molecules of the virus, antigenic property of the virus is expressed outside this lipoprotein layer. And that is called peploma. In case of HIV virus, this peploma is important. This peploma is GP120. And in case of influenza virus, the peplomas are hemagglutinin and neuraminidase. They are the binding molecule. It binds with the specific tissue or a specific cell of the hosts. Neuraminidase of influenza virus is the cutting enzyme. It is the receptor destroying enzyme. It destroys the receptors of the host cell. So these two are peplomas, and the nucleic acid, the capsid, and then the envelope is the outer layer. Nucleic acid, most DNA viruses possess double-stranded DNA except parvovirus, which have single-stranded DNA. So all DNA virus, double-stranded DNA virus. Name some DNA virus. Sir, all double-stranded, all DNA virus except parvovirus, single-stranded. Most RNA virus, single-stranded, except reovirus, rotavirus possesses double-stranded, and retrovirus possesses 2 copies of single-stranded RNA. Remember this line, retrovirus, 2 copies of single-stranded RNA. Never mention double-stranded, 2 copies of single-stranded RNA. HIV genome, please tell the HIV genome, sir, single-stranded, 2 copies of RNA. Capsid, the composed of a number of repeated protein subunits, polypeptides called capsomeres. The function is to protect the nucleic acid core from external environment, and in non-enveloped virus, initiate the step of viral replications and antigenic and specific for each virus. So when there is no envelope, this nucleic acid and capsid forms the entire virion. So in case of envelope virus, the envelope is included in the virion, but in case of non-envelope virus, the envelope is excluded from the virion particle, the infectious particle. On the basis of this capsomere, the protein subunits or polypeptides, on the basis of the different blocks of these polypeptides, they are classified into different, this is called symmetry. This symmetry, that is icosahedral and helical and complex. This is the icosahedral. 12 vertices and facets. This is facets and these are vertices. So icosahedral in nature. Complex symmetry is pox virus, helical, icosahedral and complex. Complex example, pox virus. The envelope surrounding the nucleocapsid is lipoprotein in nature. The lipid part is derived from the host cell membrane, and the protein part is virus coated, made up of subunits called peplomers. Peplomers project as spikes on the surface of the envelope. Picture is there. Nucleic acid core and then envelope. Extreme outside is the envelope. The most viruses are enveloped except non-enveloped DNA viruses are parvovirus, adenovirus and papovavirus. Non-enveloped RNA virus are picornavirus, reovirus, calcivirus, hepatitis A virus, hepatitis E virus. But how to remember? This is a different size, I will tell you. This is the size of the different size of the virus. Beautiful. Satellite-like, bullet-shaped, the different type, different shape and different virus. On the basis of the shape, the rabies virus is bullet-shaped, Ebola virus is filamentous, pox virus brick-shaped like that, adenovirus is space vehicle-like. The virus differ from viroids. Viroids is the plant virus. It's comprised of naked circular small single-stranded RNA plant virus called viroids, and prion consists of abnormal infectious protein molecules without nucleic acid. So virus particle without the nucleic acid is the prion, that is highly resistant to physical and chemical agents. The causative agents of prion disease, Creutzfeldt-Jakob disease, and viroids is the plant virus. Susceptibility to physical and chemical agents, as the outer layer of the enveloped virus, it comprises of lipoprotein layer. So that particular layer is susceptible to chloroform, ether, that is some solvent or fat. So enveloped virus are susceptible to chloroform, ether. But the best disinfectant is oxidizing agent, hydrogen peroxide and formaldehyde. During the COVID time, 1% hypo, that is the main sterilizing agent we often used in our laboratory and the COVID lab, of course. So hydrogen peroxide is the best of all. This is the list of the herpes virus, the DNA virus, and there is a huge list of RNA virus. Please note this, how to remember. The DNA virus, the mnemonic is H2AP3. H2, that means HepaDNA virus, herpes virus. A for adenovirus, parvovirus, papovavirus and pox virus. Parvo, papova, pox. So the code is H2AP3, but you have to decode it during examination time. Now the DNA virus, all DNA viruses are double-stranded except parvovirus, that is single-stranded. All DNA viruses are icosahedral except pox virus. The naked DNA virus, non-enveloped DNA virus, the mnemonic is PAP, naked PAP. So PAP, parvovirus, adenovirus and papovavirus. Please remember these things, these mnemonics. Regarding RNA virus, the naked RNA virus are PCR test or CPR. C stands for calcivirus, P for picornavirus, R for reovirus. You can call it PCR also. The enveloped virus, enveloped RNA virus, enveloped RNA segmented virus, segmented virus. The nucleic acid may be segmented. So segmented RNA virus is very important because this particular group is highly infectious. And this, the mnemonic is BORA. BORA. B for Bunia virus, O stands for orthomyxovirus, retrovirus and arenavirus. So Bora, these are mnemonic for segmented RNA virus. They envelop non-segment, rest all others, but there is a mnemonic craft, coronavirus, rhabdovirus, flavivirus and togavirus. Now the point of positive sense and negative sense. During viral replication, this positive sense and negative sense is important. They play an important role. The positive sense, that means that they can transform this particular segment of DNA to messenger RNA by themselves. They have the ability, positive sense. They are the messenger RNA. But in case of negative sense, they carry a particular enzyme with the nucleic acid, by which they can coat for messenger RNA, and from the messenger RNA the proteins are synthesized. So they are negative and negative sense. So this positive sense and negative sense, the positive sense viral RNA is similar to messenger RNA and thus can be immediately translated by the host cell. They need the host cell enzyme, and this is translated directly to viral proteins in cytoplasm, the early viral proteins. The negative sense viral RNA cannot be readily translated into viral proteins as the RNA is antisense, 5' to 3', 3' to 5'. Antisense, 5' to 3' and 3' to 5'. Antisense is 3' to 5', isn't it? So this antisense, hence they possess RNA dependent RNA polymerase with them, the negative sense. They carry RNA dependent RNA polymerase with them, and by which the messenger RNA is formed. Now replication is easy. Double stranded DNA, the positive sense single stranded RNA virus, you see, positive sense RNA virus, easy messenger RNA conversion, and negative sense, this is RDRP is attached with this. RNA dependent RNA polymerase is there, by which messenger RNA is formed. So now the replication is easy. The viral replications. The virus undergo a complex way of cell division. The steps are attachment, by which with this peplomar, the tissue tropism is there. For each and every virus, they have a selective tissue, a particular tissue. They invade that tissue. Coronavirus, they very rarely involve the urinary tract. They are respiratory specific, they are alimentary tract specific, and this much. Encephalitis virus selectively has tropism for the CNS. Orthomyxovirus tropism for the respiratory epithelium. Rotavirus for GI tract. So everywhere, each and every virus, the tissue tropism is there. So the particular peplomars is very important. Nowadays with the peplomar, they attach the host cell receptor, and then the penetration, then uncoating. After getting entry, uncoating is required. Initially the nucleic acid is synthesized, and then the biosynthesis, other step, the non-structural protein by which the capsid is formed, and during the exit, they imbibe the lipoprotein layer of the host cell in case of enveloped virus. So the maturation and finally the release. Every step is important. No attachment, immunity. Penetration, no viral infection. Uncoating, prevent the uncoating by drug, no viral replication. The biosynthesis, inhibit the structural proteins, no biosynthesis will occur. The maturation, you arrest the maturation process, no viral replication. And inhibit the release. Even in case of defective virus, the steps are not maintained. The defective virus is deficiency in during mutation, deficiency of different enzymes, deficiency of getting the host cell enzymes. The host cells are not permissive. Host cells are not permissive. Influenza virus getting entry into the alimentary tract. Epithelial cell is not at all permissive. So every factors that counts viral replication. First and most specific step is that attachment. The HIV, the viral surface glycoprotein 120 binds to CD4 molecules of the host cells. Influenza virus viral agglutinin and envelope protein bind specifically to glycoprotein receptors present on the surface of respiratory epithelium. Penetrations, phagocytosis or viromexics, receptor mediated endocytosis. Uncoating by the action of lysosomal enzymes of the host cell, the viral capsid gets separated from the nucleic acid, is released in the cytoplasm. Biosynthesis, nucleic acid, capsid proteins, enzymes are required for various stages of viral replication. Of course, the regulatory proteins. Nucleic acid replication, the DNA virus replicates, the replication occurs in the nucleus, DNA virus nucleus, except in pox virus. The pox virus which synthesizes DNA in the cytoplasm. So DNA virus replicates inside the nucleus except pox virus. RNA virus replication occurs in cytoplasm except retrovirus and orthomyxovirus. Finally, the mechanism of viral replications. The double-stranded DNA, no problem, because the conversion is possible, positive sense and negative sense. And then messenger RNA formation is easy. In case of single-stranded DNA, it acts like the messenger RNA. So first, the second strand will be, the complementary strand will form with the host cell enzymes, and then same as like double-stranded. In case of the negative sense RNA like orthomyxovirus, paramyxovirus and abdovirus, the RNA is antisense having polarity just opposite to messenger RNA. They have their own RNA polymerase for transcription, RDRP. In retrovirus, the single-stranded positive sense RNA is transformed to RNA-DNA hybrid in retrovirus. 2 copies of single-stranded RNA. So there is an RNA-DNA hybrid from RNA to DNA, complementary strand. And then with the reverse transcriptase, finally double-stranded DNA is formed which integrates with the host cell DNA. So that integration is very important. And virus, especially the RNA virus, they have to convert to DNA for the integration to the genome of the host cell. This is the picture of the story. The basic thing is that positive sense and negative sense. Assembly, the viral nucleic acid proteins are packaged together to form virus nucleocapsids and occurs in the host cell nucleus and cytoplasm that you already know. Now, the maturations following assembly, maturation of daughter virions take place either in the nucleus or cytoplasm or membranes through the Golgi apparatus or endoplasmic reticulum, and plasma membrane. The lysis of the host cells in case of non-enveloped virus and bacteriophages. The non-enveloped virus, they release with the lysis of the host cells. And in case of the enveloped virus, they acquire the lipid envelope, the lipoprotein layer of the host cells. The active phase, that is the active phase is the entry of the virus inside the host cell and release the mature virus. The time phase of that is the active phase. And the duration is 15 to 30 minutes for bacteriophage and 15 to 30 hours for most of the animal virus. Now the abnormal replication. For 10 to the power 4 to 10 to the power 8 replications, there is one mutation. So abnormal replication can occur. And when the defective assembly, when cells are infected with large number of influenza virus, the von Magnus phenomenon, the dose is high. So the cell is loaded with the influenza virus. And during the process of replications, there is an error. So the viral replications with error is the defective or incomplete virus. Capsid is not there. Genome is deformed. Anything may occur, and this will be an incomplete virus, will not be able to replicate. No progeny will be formed. So that is the inefficient replication. Insufficient also, for there is no replication. Only one virus, two viruses are formed. The huge dose to infect that is required for the viral loading dose to infect the cell is not formed. The defective virus genetically deficient, incapable of producing infectious virion, aborting infections, defect in progeny viral release. During the release, there may be defect, non-permissive host, influenza virus in GI tract, HIV in GI tract. So it is not possible. So these are cells are non-permissive cells. Abnormal replications, co-infection, some genetically defective virus can form infectious virion in presence of a helper virus co-infecting the same cell. Liver cell, say for example. Hepatitis D virus enters the liver cell. But that virus is replication deficient virus, incomplete virus, cannot replicate. Until and unless there is hepatitis B virus infection. As soon as there is hepatitis B virus infection, the co-infection hepatitis D virus now can replicate with hepatitis B virus, never alone. So this is a classical example of co-infection of virus. The viral genetic modifications, mutations I have mentioned, and the mutants occur spontaneously and may be induced chemically, or by physical agents, UV light, etc. The types of mutations, one is a conditional lethal mutant. Already you know that is temperature sensitive. The type of conditional lethal mutant that can grow at a low temperature at 20 to 30 degrees centigrade, but not at higher temperature at 37 degrees centigrade. On the basis of this, we have prepared a vaccine for respiratory tract disease such as orthomyxovirus vaccine and coronavirus vaccine. In Antennae and Naris, the temperature is low. They can survive there, they can multiply there, but as soon as they enter the respiratory tract, the temperature is high, so they are destroyed. But the antigen produced there and the virus can grow over there on the Antennae and Naris with this property, that is the temperature sensitive mutant, by which we can arrest the coronavirus in frank cases, wild virus. So interactions between viral genes, when two or more virus particles infect the same host cells, that is called viral interactions in genetic and non-genetic. Two concepts, one is viral interference, when two viruses infect a host cell or cell line, Sometimes leads to inhibitions of one virus called viral interference. One particular species of virus can interfere the growth or replications of other viruses. Interferences does not occur with all viral combinations, and many viruses may infect and multiply together in a host cell. Oral poliovirus is a classical example where viral interference is seen. OPV serotype interfere with the spread of wild poliovirus. That's why OPV is till date we use this OPV virus, and thus played a crucial role in control of polio outbreak. The viral genetic interactions, the transfer of genetic materials between related virus called genetic recombination occurs between two active and inactive virus or one active and other inactive virus occurs in dsDNA and some RNA virus. So, genetic material transfer, say for example, this is a host cell, two related virus enters the same host cell. Now, the genetic property can be interchanged. This is called genetic recombination. Genetic recombination. And in case of reassortment, two segmented virus, RNA virus, it enters the same host. A new hybrid virus or novel virus can be produced. You have heard the name of novel virus, coronavirus, the new strain of coronavirus, influenza virus, new strain H1N1 is a novel virus. So, how the novel virus they are formed? Because of this thing. They are segmented virus. In case of coronavirus is not segmented, but this influenza virus, orthomyxovirus is segmented. 8 segments are there. So, there is a combination can occur inside the cell between the segments, and a new virus or hybrid virus, novel virus called is formed. So, this particular segmented virus is responsible for forming the novel virus. Marker rescue, another property, reactivation, where epidemic strain of influenza grows slowly in embryonated egg. The marker rescue is that when dead virus existing inside one dead virus, one live virus enters, and that dead virus can get some unique properties from the live virus, and now it is activated. This is called marker rescue, another property of viral infection. The viral infection, the pathogenesis is that as soon as the virus enters the host cell, there is a viremia, and with this viremia, the virus enters the different cells in the bloodstream and to the bloodstream, from the bloodstream to different tissues, and now they are getting their specific tissue from the bloodstream spread, and now can infect the particular tissue. Now, during the process, the cell, the host cell can be lysed, can be transformed, and can acquire the new properties. And this is practically, if you say the pathogenesis, then it depends on the immune response of the host cell also. So, if the antibody has minimum role, and as it is intracellular, the cell mediated immunity, that plays the highest level of try to inhibit the infection through the cellular mechanism, the cell mediated immunity. And during that first inhibition is by interferon. The different type of interferons are produced, and that produce an antiviral environment and try to inhibit the viral replication. The cell may be lysed, may be transformed, may acquire the new property, new antigen, and some oncogenic virus there, they can transform and can immortalize the cell. In case of some herpes virus, the cells are immortalized. So the pathogenesis practically depends on the immune response, and inclusion body is the aggregates of virion or viral particles that are isolated, that are seen inside the host cell, and even we can identify them with a microscope. The characteristics of the special viral infections, and they have distinct size, shape, location and staining properties by which they can be demonstrated in viral infected cell under light microscope. Various routes are there. The host cell viral interactions, initial viremia, virus settles in the target station, replication, there may be cell death, the tissue damage. The virus cell beat up the host cellular immune response. The interaction can result in cell death, cellular proliferation, malignant transformation, as I have mentioned, and steady state of infection, the peaceful coexistence. The outcomes of viral infection. There may be a full-blown acute infection. This when there is cell lysis, tissue damage and resultant pathology. Viral disease, well-established viral disease can occur. If immune cells fail to protect us, then there is a full-blown acute infection of the viral disease. The chronic infections, after full-blown disease, some virus in cytoplasm may persist. It is intracellular, so can persist in cell cytoplasm. Low level, the viral load is less. I have mentioned in the earlier phase of my lecture, the viral load is less. Few viruses persist. There is a compromise. The latent infection is the dormant state. Viral load is there, but the dormant stage may activate in presence of any trigger. Subclinical level, they persist. No clinical test. By no clinical test, you will be able to understand. You will be able to identify the disease. It persists at the low clinical level. The latent phase. Intermittent exposure. Intermittent, whenever the immunity goes down, they will proliferate. Herpes virus infection, herpes labialis, যেটা লিপে হয়, যখন ইমিউনিটি কম হয়, যখনই জ্বর হলো, herpes labialis, ট্রাইজেমিনাল নার্ভে থাকে, স্যাক্রাল প্লেক্সাসে ঢুকে থাকতে পারে, এগুলি। The latent infection and cytopathic effect. Cytopathic effect, the cell is distorted. The cell acquires a special feature. The nucleocytoplasmic ratio is altered. The adhesion property, one virus adhered with other virus, and inclusion bodies, of course. The examples of inclusion body. Inclusion body may be intracytoplasmic inclusion, maybe intranuclear inclusion. We often ask, name some inclusion body which are cytoplasmic. Name some intranuclear inclusion bodies. So this is important for you. Example of intracytoplasmic inclusion body is Guaneri body, vaccinia virus, neovirus, and rabies virus is Negri body. The intranuclear inclusion in herpes virus, respiratory syncytial virus. Cowdrey body. The persistent viral infection is characterized by viral infection without cell death. This may be non-lytic, chronic, latent, recurrent, transforming. HSV typically causes a latent infection in neuron that lacks the machinery to transcribe the immediate early viral genes essential for viral replications. The host response to viral infection. What will be the response of host cell? How will you produce the first defense mechanism? This is the first defense mechanism of the body, that is known as the interferons. Interferon of host cell origin, not of viral origin. This is host cell origin. The interferon was first noticed by Isaac, and in 1957 interferons are heterogeneous and complex group of glycoprotein produced by cells and induction of infection by viral or non-viral microbes, but they show antiviral activity by inhibiting the viral protein synthesis, and it is the natural defense mechanism. The special thing is that the host cell produce a protein known as TIP, that is translation inhibiting proteins. Translation inhibiting protein is synthesized within the host cells, and this TIP is a mixture of three different enzymes. One is the protein kinase, other one is the RNase and oligonucleotide synthetase. They all together block the viral RNA synthesis and propagation of virus from one cell to another cell. They produce an antiviral environment surrounding the viral infected cell. So only they produce, they are not antiviral, they cannot destroy virus, but they can produce an antiviral environment surrounding the cell involved by the virus by producing this mixture, TIP. The types: interferon alpha, interferon beta and interferon gamma. Interferon alpha, interferon beta and interferon gamma. Alpha is non-glycosylated protein produced by leukocyte. How to remember? The beta, a glycoprotein, the same thing. It is glycoprotein. The first one is non-glycosylated. What is the difference between glycosylated and glycoprotein? Glycosylation and glycoprotein. Chemistry te porecho? Pore din bolo. The glycosylation and glycosylated and glycoprotein molecule produced by fibroblast. And interferon gamma is a glycoprotein molecule produced by T lymphocytes or NK cells. So the mnemonic is LFT. How to remember? LFT. The source to remember is LFT. Alpha, leukocyte, beta, F, fibroblasts, and the last one, gamma, from T lymphocytes. Okay? The clinical applications in chronic hepatitis B and C virus to prevent risk of cirrhosis and hepatocellular carcinoma. They are the causative agents of hepatocellular carcinoma, hepatitis B and C. Interferon with ribavirin has proved to be more effective than interferon alone for treatment of Hep C virus. And currently interferon is also used for papillomavirus, condylomata acuminata and multiple sclerosis. Now the laboratory diagnosis. The lab diagnosis, initially the purpose is to start the antiviral drugs, whatever we have. Screening of blood donors for HIV, hepatitis B, hepatitis C, that is important to stop the propagation. The surveillance purpose for outbreak of epidemic investigations and to start the post-exposure prophylaxis. The direct demonstrations of virus by electron microscopy, immunoelectron microscopy, fluorescence and light microscopy, detection of viral antigens by various formats such as ELISA, direct immunofluorescence test, immunocube test or what is the ICT? What is ICT? Huh? Flow through assay. Immunofluorescence, lateral flow through, তোমরা দেখোনি lateral flow? দেখোনি? দেখানো হয়নি? HIV তে করি আমরা কার্ড টেস্ট, ক্যাসেট টেস্ট ছোট ছোট, দেখোনি এখনো? এবার ভাইরোলজিতে দেখবে। The detection of specific antibodies. The conventional techniques such as HAI, hemagglutinin inhibition test, neutralization test, complement fixation test, newer diagnostic formats as ELISA, ICT, flow through, molecular methods of to detect the viral genes, এই সমস্ত PCR গুলো, different real time PCR and multiplex PCR etc. The isolation of virus, animal inoculations, embryonated egg inoculations and tissue cultures. এখন আর কোনো কিছু করা হয় না, কন্টিনিউয়াস সেল লাইন কালচারটা করা হয়। এগুলো পরে একবার আমরা বলবো, তখন সুবিধা হবে, এখন বুঝতে অসুবিধা হবে, পুরো ভাইরাসটা তো পড়া হয়নি। ইনক্লুশন বডিজ ধরবার জন্য হিস্টোপ্যাথোলজিক্যাল স্টেইনিং করা যেতে পারে। At the detection of viral antigens, various formats are available for detection of viral antigens in serum and other samples. Some important antigen detection tests include Australia antigen, HBsAg, hepatitis B virus কাজে লাগে, HBeAg antigen detection for hepatitis B, NS1 non-structural antigen detection for dengue virus infection from serum. এই non-structural antigenটা নিয়ে পরীক্ষা ও প্রশ্ন করতে শুনেছি আমি এক্সটার্নালদের। Non-structural, এই non-structural antigenটা কিছু নয় বাবা, দিস ইজ সিম্পলি, এটা কি করে, দ্য হোস্ট সেল মেকানিজমে যখন হোস্ট সেলে ঢুকলো তখন হোস্ট সেল মাল্টিপ্লিকেশনসটা যাতে না হয়, হোস্ট সেলকে তার এনজাইমেটিক ইনহিবিশনটা এটা করে নন স্ট্রাকচারাল প্রোটিনস। ভাইরাসের স্ট্রাকচারাল অ্যান্টিজেন আছে, নন স্ট্রাকচারাল আছে। এই নন স্ট্রাকচারাল যে এনজাইমগুলো বা নন স্ট্রাকচারাল অ্যান্টিজেনগুলো ইনহিবিট করে হোস্ট সেল মাল্টিপ্লিকেশনকে, হোস্ট সেল যাতে আর মাল্টিপ্লাই করতে না পারে সেটা বন্ধ করে, কারণ ও তো হাইজ্যাক করবে হোস্ট সেলের এনজাইমগুলো নিয়ে নিজের মাল্টিপ্লিকেশনে কাজে লাগাবে, দ্যাটস ওয়াই ওটা ইনহিবিটরি এফেক্ট আছে একটা। এটাই হচ্ছে নন স্ট্রাকচারাল অ্যান্টিজেনের কাজ। P24 অ্যান্টিজেন ডিটেকশন ফর HIV ফ্রম সেরাম। এই P24 এর অ্যান্টিজেন হচ্ছে আর্লিয়ার টেস্ট টু ডিটেক্ট HIV। একটা HIV এক্সপোজার হলো পেশেন্টের, তার সেরো কনভার্সন হয়নি, মানে অ্যান্টিবডি তৈরি হয়নি, সেই সময় অ্যান্টিবডি টেস্টগুলো করলে পজিটিভ আসবে না, ওই ল্যাটারাল ফ্লো থ্রু টেস্টগুলো আইসিটি এই সমস্তগুলো করলে আসবে না পজিটিভ। কিন্তু তখন যদি 4th জেনারেশন টেস্ট আসে যার মধ্যে P24 অ্যান্টিজেন থাকে, এইচআইভি অ্যান্টিজেন, সেটা যদি ডিটেক্ট করা যায় তখন এইচআইভি ইন আর্লিয়ার স্টেজেস ধরা সম্ভব। এখন এই P24 অ্যান্টিজেন টেস্টটা এসছে। আর হচ্ছে এমব্রায়োনেটেড এগ, হচ্ছে অন্তত 12 দিনের এগ আমরা নেই, 12 days এমব্রায়োনের, মানে এমব্রায়োনেটেড এমব্রায়ো তৈরি হয়েছে এমন ডিমের আর কি। তার মধ্যে অ্যামনিওটিক ক্যাভিটি, কোরিও অ্যালান্টোইক মেমব্রেন, অ্যালান্টোইক ক্যাভিটি, অ্যানিমব্রিও, এই চারটের মধ্যে আমরা ভাইরাসকে ইনকুলেট করি। কোরিও অ্যালান্টোইক মেমব্রেনের মধ্যে আমরা হার্পিস ভাইরাসকে দিতে পারি, বা এনকেফালাইটিস, জাপানিজ বি এনকেফালাইটিসকে দিই। হেমারেজিক পক্স হয়, পক, ছোট 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 পক হয়। আর ভাইরাল কালচারে আমরা সেল লাইন কালচারটাই এখন ইউজ করি বেশি, তার মধ্যে হেলা সেল, হেপটু সেলস এইগুলো এখন আছে, যেগুলো সবই কার্সিনোমার প্রপার্টি ক্রিয়াকে নেওয়া হয়েছে, মানে অ্যান্টিজেনিসিটি অফ কার্সিনোমার অ্যাভয়েড করা হয়, কিন্তু প্রোপাগেশন, মাল্টিপ্লিকেশন, কন্টিনিউয়াস মাল্টিপ্লিকেশন প্রপার্টি অফ দ্য কার্সিনোমা সেল, সেটাকে কিন্তু মেনটেন করা হয়। তাহলে উইথ দিস প্রপার্টি কার্সিনোমার মাল্টিপ্লিকেশন প্রপার্টিকে রাখা হলো, আর তার অ্যান্টিজেন যেগুলো যেগুলো কার্সিনোমেটাস অ্যান্টিজেন, সেগুলোকে ডিস্ট্রয় করা হয়। নিয়ে একটা সেল লাইন তৈরি করা হয়, মানে টিস্যু দেওয়া হলো, টিস্যুর মধ্যে সেগুলো টিস্যু যেগুলো সেল কালচার যেগুলো, তার মধ্যে টিস্যুর মধ্যে আমরা ভাইরাল প্রপার্টিগুলো, ভাইরালগুলোকে ইনকুলেট করি, তখন টিস্যুর মধ্যে গ্রোথ হয়। সেগুলো দেখবার অনেকগুলো রাস্তা আছে, সেইভাবে করি। একটা সেল ভাইরাল কালচার বলে আছে, সেটাও খুব ভালো। এটা নিয়ে আমরা পরে আলোচনা করব, ভাইরাস কালচার কি করে করা যায়, এমব্রায়োনেটেড এগ কালচার, সেগুলো নিয়ে আমরা আলোচনা করব পরে। আজকে এই অবধি হলো, আর অ্যান্টিভাইরাল ড্রাগস গুলো নিয়ে পরে আলোচনা করব।